I'm official.
Throughout the 2018-19 academic year, the PPMS match team has been researching on Aquaporin Z, an aquaporin present in E. coli. Take, for example, the story of Pajit, which faces hardships in finding clean water in India. This leaves them with a high risk of contracting waterborne diseases, and implementing biomimetic water filtration in India can solve India's water crisis.
This is not only a water problem, it's also a sanitation problem. This could be due to the fact that only 33% of Indians have access to traditional sanitation. And the future for the water crisis is just going to continue to get worse. By 2030, water scarcity will displace 24 to 700 million people. By 2040, there will be no water in most parts of India. And by 2050, India's projected population will rise to 1.7 billion people.
So, to solve this dilemma, our MAPS team decided to implant aquaporins into an artificial membrane. So, as you can see here, Aquaporin Z consists of mainly 6 helices and 2 half helices, and they form a tunnel which strips away any excess materials from water molecules like hydrogen ions, bacteria, salts, etc. So we decided to use E. coli because it's easily found in nature and it's also easy to harvest. And research has shown that it's very effective as a water filtration method.
So for our motto, we decided to focus on, first, the aromatic arginine selectivity filter. So the aromatic arginine selectivity filter, the aromatic portion of it means it's kind of in a ring shape, just like phenylalanine is here. So the aromatic arginine selectivity filter is the smallest and the narrowest pore, and it has a very narrow diameter, removing all the excess material. And the aromatic portion, the ring portion of it, just makes it even smaller, therefore guaranteeing even more of the water is going to be purified. So as part of this, we have phenylalanine. This is hydrophobic, so it's a side chain of the amino acid that forces water molecules to go through the pore by repelling them down the pore. We also have histidine and threonine. They're hydrophilic side chains of the amino acid that bond to each water molecule. And we also have arginine, which is a positively charged amino acid that repels hydrogen ions. So again, there are no hydrogen ions, again, purifying the process. We also focus on the MPA motif, which is an asparagine proline alanine sequence. We decided to feature the asparagine portion of it, which are on the half helices, yellow and orange. So you can see them through here. So this is featured because it's the hydrophilic part of the MPA motif, so it'll attract water and then push them down the tunnel. So this again allows for more efficient and selective filtering. And also, the gray outlines here, we can see that this is kind of the channel, and the blue loops are what connects them. So this is our motto.
This is the results. So this is a graph comparing 3 different types of aquaporin, aquaglyceroporin, aquaporin 1, and aquaporin Z. Aquaglyceroporin, as you can see, has the highest pore diameter because it lets in both glycerol and water, while these 2 aquaporins have smaller pore diameters that let in only water.
As you can see here, this is a graph of water productivity, which is defined as permeability per unit of driving force. So these are different methods of commercially available water filtration, such as forward osmosis and reverse osmosis. And also, the higher the number, the better or more efficient the water filter is. This is a membrane without the implantation of aquaporin, and this is a membrane when aquaporin is placed into the membrane. And as you can see, aquaporin implanted membranes rank way higher than any of the other commercially available water filters. And this is why, in conclusion, because of its small pore diameter and aromatic arginine selectivity filter, Aquaporin Z is the best protein for the job.